Welcome, folks. Today we are discussing about the complex inter-organ crosstalk between skeletal muscle and liver metabolism during starvation. And that sounds fascinating, but before we get into the details, why exactly is that link so important? I usually think of the liver as the main hub for lipid storage. Well, that's exactly the point. Traditionally, we look at the liver in isolation, but this research highlights how skeletal muscle, the largest metabolic organ in our body, is essentially calling the shots for systemic health. The study focuses on a family of transcription factors called FoxO. Right, I've heard of FoxOs as regulators of aging and stress. Are they just in the muscle? They're found across tissues, but we're looking specifically at their loss in skeletal muscle. Now, here is where it gets counterintuitive. When these mice were starved for 48 hours, they didn't exhibit the usual muscle wasting. You'd think that's a positive, right? I see, interesting. So the muscle was protected? That seems like a good thing, but I have a feeling there's a downside. Exactly. While their muscle stayed robust, those same mice developed severely exacerbated hepatic steatosis. That's the accumulation of fat in the liver. It turns out that by preventing muscle atrophy, they essentially crippled the muscle's ability to help the liver process lipids during energy deprivation. Got it. So it's a trade off. How does the muscle actually talk to the liver to manage that lipid load? It comes down to gene regulation. The study found that FOXO proteins are responsible for turning on critical genes in the muscle, responsible for triglyceride uptake and fatty acid oxidation. Without these proteins, the muscle can't properly burn fuel or manage lipids, and the liver ends up bearing the entire burden. I have to challenge you on that for a second. If the muscle is hoarding its fuel instead of burning it, why doesn't that just help provide more energy for the rest of the body? That's a sharp observation. The problem is that the burn process, or fatty acid oxidation, is the key. Without functional FOXO signaling, the muscle loses its ability to utilize fatty acids efficiently. This backfires systemically. Plus, these mice showed abnormal glycogen accumulation in the muscle, suggesting their internal metabolic machinery is totally out of sync. And what about the other side of the coin? What happened when they increased FOXO activity? That was the most exciting part. When they overexpressed FOXO1 in skeletal muscle, it actually protected the mice from metabolic dysfunction associated steatohepatitis, or MASH, even when the mice were fed a harsh Western style diet. It increased an enzyme called ATGL, which helps break down triglycerides. Essentially, the muscle became a much more efficient lipid manager. So, effectively turning up the muscle's metabolic dial can actually fix a liver problem. Precisely. It showcases that skeletal muscle acts as an endocrine like communication center. It's not just a passive tissue, it's actively regulating whether your liver becomes fatty or stays healthy. This really changes the perspective on how we approach metabolic diseases like MASLD. It's incredible that such a specific genetic mechanism can have such wide reaching effects on the whole body's lipid homeostasis. Absolutely. It bridges the gap between basic cell biology and systemic pathophysiology. We've barely scratched the surface, but this provides a clear target for future therapeutic research. Well, that's all the time we have for today. Thanks for joining us for this deep dive into the hidden world of muscle liver communication. Goodbye, folks.